All right. Welcome everyone to the DDPS seminar. Before we introduce our invited speaker, let's go over some rules and logistics. First, please mute yourself during the talk, but if you have questions, you are welcome to unmute and ask questions, or use chat room to post your questions so that we can address them in Q&A session at the end. Second, today's DDPS seminar is open to external audience, therefore no classified discussion is allowed. Finally, the talk today will be recorded and uploaded in our YouTube channel. Okay, let me briefly introduce our speaker today. It is an honor to host Jan Christoph, who is an assistant professor at cardiology and bioengineering in University of California, San Francisco. Please close by from Livermore, where I am. He also serves as head of cardiac vision laboratory in UCSF. Jan obtained PhD in biophysics at University of Göttingen in Germany in 2014. He did his postdoctoral study at Max Planck Institute for Dynamics and Self-Organizations from 2014 to 2018, and served as a staff scientist at Department of Cardiology in University of Medical Center Göttingen in Germany before he came to UCSF. He has earned NIH New Investigator Award, DP2, in 2022. Okay, today Jan will talk about predicting heart rhythm disorders from spatio-temporal imaging data using artificial intelligence. Please enjoy and expect a wonderful talk. Now let me ask a couple of questions, if you don't mind, Jan, just to get to know you better and then make this seminar somewhat informal. The first question is, obviously you are passionate about heart or medical field in general. Is there any moment in your life when you realize that, wow, this is it, this is the field I would like to commit my career? Do you have that moment? Yeah.
Sorry, the question was, when was that moment when I wanted?
Yeah. And if you can describe that moment.
Yeah. I think one thing that I need to say before is that I never wanted to become a scientist. It was not a lifelong dream of mine when I was young. I never imagined I would be a professor or a scientist later on in my life. I think I did a PhD out of curiosity because I wanted to spend time on working on quantitative biology. I think that was my interest somehow. And then spending 6 or 7 years, or almost 8 years on this one important publication that defined my career, that came out in 2018 in Nature, even opened up, I think, the way for me to even consider becoming a professor. So it was a pretty late decision. So I was already way into my postdoc and published an impactful paper, and then realized, oh wow, I can actually do this if I want to. I can try and get a tenure track faculty position and keep doing this for the rest of my life, maybe even. And I think before that, I didn't have necessarily the plan to do that.
I see.
So it was, I think, at that point, I was definitely hooked. I was really intrigued by this type of research that had built up over the years, and then in that moment it became clear, okay, I want to do that. That was roughly in 2018.
Wow. I don't know, at some point in your presentation today, maybe you can share what was that paper?
Yeah, I will have one slide about that paper, and it's
awesome.
It is a kind of the foundation for the second half of my talk, this paper.
I see. Well, okay. I have one more question, if you don't mind. Obviously, you're using artificial intelligence for your research. When were you first exposed to artificial intelligence and decided to use that in your research?
So I think the first time I heard about convolutional neural networks was in the cafeteria at Max Planck in 2012, 2013, when AlexNet and all that came out and people talked about it. But for many more years, I didn't get my fingers on it.
Yeah.
And it was actually only during the pandemic when our institute shut down, when I sat at home and my student and I, we both kept talking about how we wanted to progress and make, improve certain computational methods that we had developed, and talked about autoencoders and UNet. And I just tried it out, and it worked right away, and it was amazing. And I will have in my talk, I will have a few slides about exactly that.
Awesome, awesome. Thank you, Jan. Our stage is yours.
Okay, thank you, Ying-Su, for the invitation and the introduction. Again, my name is Jan Christoph. I work at the Cardiovascular Research Institute. I'm appointed in the Division of Cardiology here at UCSF. My background is in physics. I obtained a PhD in physics, and let me briefly introduce my lab. And you will notice here that most people in my lab also have a quantitative background, either computer science, physics, or electrical engineering. And the pictures here are kind of weighted by how many contributions or how important these lab members are or were over the past years. And you can notice here Jan Lebert is a central figure in my lab. He was also a PhD student in Göttingen in Germany, and came then with me to set up the lab here at UCSF. And a lot of the work that I will talk about today was actually done by him. Then we were also able to recruit Shrey last year, who has a background in physics and computer science, worked as a software engineer, and he is interested in computer vision. So he has built important numerical methods for us. And then on the right side, Daniel, he is now at Caltech. He was an undergraduate student who made very important work last year together with Jan. And on the bottom left here, Tanish, who has an interest in, everything deep learning and in particular diffusion modeling. And he did the diffusion modeling work that I will also show. So what can we do as physicists, mathematicians, or engineers in cardiology? And if you are interested in this topic a bit further, I can recommend this podcast that came out recently in Quanta magazine. That's a really nice magazine I often read. And Stephen Strogatz, who's an applied mathematician from Cornell, he interviewed Flavio Fenton, who's a colleague of mine from Georgia Tech. And he, since many years, leads a group there, and they research questions such as how can we predict what the heart does next, and how can we intervene and apply defibrillation pulses in order to stop heart rhythm disorders. And they talk about that at length. And we do very similar research with a slightly different focus. We try to focus on imaging itself, and we try to understand the interplay between mechanics and electrophysiology in the heart. So our community is small, but very persistent and very active, and very interdisciplinary. We have, cardiologists with an interest in machine learning who also have a background in engineering. We have mathematicians, applied mathematicians, engineers, physicists, and they all have their different views and approaches. But we together, we meet quite frequently, at least once a year. This was, for instance, a meeting in 2018, which also, I think, helped me to make this decision that I want to stay in academia and that I want to give it a try to keep pursuing these projects and these research topics that I'm currently working on. It was at the KITP at Santa Barbara. And I would say that these people like us who have a quantitative background are a very small group in this huge arena of cardiologists and biologists and physiologists who research heart rhythm disorders or heart disease in general. And there's roughly an even distribution between, mathematicians, physicists, engineering, computer scientists. Hard to say who dominates this field, I would say. Last year, we organized a meeting or a mini symposium at a SIAM conference, and this year again we will meet at a SIAM conference. So this is also something that we often participate in or organize. And the people who come there are regulars, people who've come to these meetings since many years. They keep showing up. So the heart is an electromechanical pump. That is something that I need to introduce first. The most important property of cardiac muscle cells, and you see here one on the left side, is that they are electrically excitable. Their transmembrane potential undergoes variations over time. That's a way of communicating for these cells. These changes are referred to as action potentials. You see there on the right side, 3 curves. The first one, the green one, is the action potential. As soon as a cardiomyocyte experiences such an action potential, it opens intracellular storage for calcium. And the calcium is then released into the cell, and the calcium kind of fuels the contraction of the cell. So the contraction follows the action potential, is elicited by the action potential. Then this electrical excitation overall on the global scale in the heart triggers and orchestrates the heartbeat. We have in our heart multiple centers, the sinoatrial node and a backup node, and in there are cells that beat autonomously roughly 1 time per second. And then they shoot out electrical excitation through a cable and then through a branch of other cables into the main heart muscle tissue that is shown here. And this heart muscle tissue consists of roughly 5 billion cardiomyocytes. And once this electrical excitation reaches the end of these so-called Purkinje fibers, they exit at these sites, excite the first cardiomyocyte or a small group of cardiomyocytes, and they pass on this excitation to their neighboring cells. And so via a diffusive process, this excitation then is propagated through the entire cardiac muscle. And this excitation propagates through the muscle tissue at a speed of roughly 1 meter per second. So it's relatively fast. Then atrial fibrillation and ventricular fibrillation are the 2 most severe forms of heart rhythm disorders. There are many different flavors of heart rhythm disorders, but those are the most prominent ones because they either affect many, many people or they are highly deadly. So atrial fibrillation affects the prechambers of the heart, the atria. That can be a chronic condition that you can have for many weeks or months before you have your elective procedure to get rid of atrial fibrillation. Atrial fibrillation itself is very problematic because it can cause heart failure or induce ventricular fibrillation, which then affects the main chambers and is immediately life-threatening. However, ventricular fibrillation can also occur on its own without atrial fibrillation. It can occur spontaneously, and you often have, sadly, these reports here I can update this slide every 3-4 months. Another celebrity dies from sudden cardiac death. It is very common, and it affects really people at all ages. It can sometimes affect people who do sports, especially in professional sports. At a very young age, it can affect people with genetic defects at a young age, but of course, it becomes more and more prevalent the older you become. Physicists describe these electrical wave phenomena with partial differential equations. I will have a few slides about that, and here are some reviews about that, and which nicely summarize the work that has been done in our field. In short, you can see here an electrocardiogram at the top that describes a sudden onset of ventricular fibrillation, so the most severe and most deadly form of a heart rhythm disorder. On the left side, you can see the QRS complex here, which corresponds to a heartbeat. This here corresponds to the atria contracting, this to the ventricles contracting, and you see 3 heartbeats before all of a sudden, out of nowhere, this ventricular fibrillation sets on. And we associate with this episode here 3 computer simulations that describe the spatio-temporal dynamics of the electrical waves that determine the heartbeat during these 3 stages here. So the normal heart rhythm is caused by a plane wave that really originates down there where the Purkinje fibers inject the excitation into the heart muscle. Then this wave propagates upwards and needs to be reinitiated by the sinus node. And here, what happens here is that this wave becomes perturbed and undergoes a transition into this spiraling or rotating state. And we refer to this wave form here as a scroll wave. A scroll because it's curled up like that. And this scroll wave can even further degenerate into spatio-temporal chaos, which is shown here on the right side. And these computer simulations are supposed to illustrate what we think happens inside of the heart muscle during these different stages. We can model cardiac cells in that sense as relaxational oscillators or nonlinear oscillators. And you see here an example of a quite old mathematical model for such a relaxational oscillator, the Fitz-Nagumo model. And in such a model, you have two equations. One stands for the excitation, one for the refractoriness, and they kind of compete with each other. Whenever the excitation goes up, the refractoriness also goes the excitation. And in phase space, you then have this behavior here that most of the time the cell is in the fixed point, but if you perturb it slightly, if the cell receives excitation from one of the neighboring cells, then you perturb this oscillator here over this separatrix, and you see an excursion through phase space, which corresponds over time to an action potential. This mathematical modeling of this electrical activity can in principle be divided in either this synthetic bottom-up approach, where you measure all ions in cells or ion channels and their contribution to the cellular transmembrane potential and ion fluxes to the cells, or an analytical top-down or phenomenological approach, such as the Fitz-Nagumo approach, where you kind of mimic the behavior of action potentials using these relaxational oscillators. And of course, over the years, this was back then in the 50s, many different models were developed, and we kind of distinguish them into either these ionic, very detailed models, which are computationally expensive, or phenomenological models, which are cheap and easy to implement, but also are less realistic, or any hybrid version between these two extreme forms. And this is kind of the sweet spot. For instance, the Fenton-Karma model is one model is used widely, and there are many other models which consist of fewer equations and fewer parameters, but sufficiently many parameters so that you can recapitulate the most important dynamics of the heart. So if you then connect all these oscillators in a tissue, you have then nonlinear waves of electrical excitation, which propagate through the tissue. And here, this is a cellular automaton. And you see if you excite one cell, it passes on the excitation to neighboring cells. And eventually, this wave dies out at the boundary because it's followed by this refractory wave that prevents further re-excitation of the tissue. And this is important. This is the most important property of our heart, that the excitation is supposed to occur once a second, and that it's also supposed to die out so that the heart can relax again. The big analogy is, for instance, a fire front. You have excitable tissue that is maybe dry grass. The excitation wave itself is a fire front, and the refractory part is the fuel has burned and is gone, and the fire can turn around and re-excite or reignite the forest. Now, in a continuous medium, we can nicely simulate this with PDEs. And in this case, this is a special form of a reaction-diffusion system. You can see here again, this is a diffusive term. These are local reaction terms that describe the excitability of the tissue. So again, you have a rest state and a threshold. And once you perturb this oscillator over this threshold, you have this excursion, the action potential. And then due to the diffusion, you have here this wave that propagates outwards and then dies out at the system boundaries. Now, cardiologists, they try to measure these wave phenomena in patients. However, there are many limitations in terms of how practical it is to access the tissue, to measure and obtain recordings from patients. The gold standard currently is to use so-called catheter mapping. So catheters are inserted into a patient. This procedure is called minimally invasive, but nevertheless, it is invasive. And then under anesthesia, cardiologists record here electrical activity from these tips of these catheters. And there are many different designs and shapes, but what it is at the end of the day, it is a point-by-point measurement, and it's a lengthy measurement. It's time-consuming because they have to reposition the catheter, and you have to assume that this rhythm that this patient is in is stable. As soon as it is irregular and chaotic, they cannot obtain reliable measurements. So it is really only applicable in patients that have a stable abnormal rhythm. Nevertheless, you can obtain very nice maps from patients. This here is a patient that is in a procedure and has ventricular tachycardia. You can see here two maps. One was obtained at the epicard, the outside of the heart, and one was obtained on the endocard of the heart on the inner surface. And these two recordings were obtained kind of simultaneously and are synchronized in time. And you see here this kind of rotating wave pattern. And this is a wave pattern that shouldn't be there, that is abnormal, and that causes irregular beats or irregular contractile function. And it's circulating around this area here where there's presumably a scar in this patient. So this patient had a myocardial infarct. And then what can happen is that these waves get caught there and they kind of circulate around these infarcts. However, there can also be much more complex rhythms. And you can see here an excised PIC heart on the left side that was excised at the Maastricht University in the Netherlands. And you can see here very nicely how chaotic this heart contracts during ventricular fibrillation. And on the right side here, this is a rabbit heart that we have imaged in our lab during ventricular fibrillation that we induced electrically. And I will slow down this video for you now, and I hope it plays smoothly. You can see now overlaid action potential waves in pink, which correlate with this mechanical contraction that the heart exhibits during ventricular fibrillation. And so here we have a measurement of these chaotic wave patterns obtained on the surface of the heart. Another example, how fast and how irregular the contractions can become. And this is a very extreme example of a PIC heart fibrillating. There's almost no contractile activity anymore. And this is why ventricular fibrillation is so dangerous. There's no blood flow any longer. So we can, in a laboratory setting, excise hearts and keep them alive for roughly 1 day and do imaging experiments with these hearts. We perfuse them with a liquid called Tyrol solution, which is essentially water plus salts plus sugar and oxygen. So we keep the pH level at a level where it's supposed to be. And then we apply something that is called retrograde perfusion. We pump blood, this solution here, retrogradely into the heart. It closes the valves and it goes into the coronary vessels and perfuses the heart, and therefore can be kept alive for many hours. So this is our imaging setup that we have built here at UCSF. You can see the centerpiece is here, this imaging chamber. Inside of that chamber is this liquid and the intact heart. The heart can also be seen here in 12 video images. These images are acquired simultaneously at very high speeds, 500 images per second, by these cameras that are here on this table. And on the left side, you can see a commercial ultrasound system that is also integrated into this imaging system. And this ultrasound system records three-dimensional volumetric images of the heart. I hope this plays well. You can see here a rabbit heart beating in our tank. This is the same rabbit heart, and this is the ultrasound transducer here at the bottom. It's a 2D matrix array transducer that produces between 25 and 100 volumes per second. Again, this is a drawing here of these different camera images. These cameras are calibrated, and we were able to reconstruct the entire surface of the ventricles with 12 cameras that are synchronized and calibrated. And then we cross-register the optical reconstructions that we can make with the cameras with the ultrasound data, so that we have structural imaging data plus functional imaging data that we acquire optically. Because optically we do not only image the surface of the heart, but we add fluorescent dyes, and these fluorescent dyes are then excited with this green light on the left side. You can see here 48 LEDs that are also integrated into this imaging setup, and they illuminate the heart from all sides evenly and excite fluorescent dye, which then emits a particular wavelength, more in the red. And this emission light then carries the information that we can measure and from which we can extract action potential waves. And this here now is one such measurement during the normal heartbeat, sinus rhythm. You can see these are 3D reconstructions, relatively high resolutions, the heart contracts. And you will also notice, if you watch closely, that there's a slight decrease in intensity. I will slow this video now down. Again, we have added voltage-sensitive fluorescent dye, and this dye indicates action potential waves or voltage changes. I don't know if you can see that on Zoom. You may or may not see it. I can see it pretty well on my screen. There's a slight decrease, and I will now amplify this decrease. This is the routine processing step that we do. We extract the signal by simply amplifying it over time. And you can see here the action potential wave front in black. So this activation occurs in multiple sites in the ventricles due to this Purkinje fiber system that ejects this excitation in several sites. We can also measure then accordingly mechanical strain. What you can see here is the area change, and in red that is a decrease in area change, and blue is a dilation or increase. So that we are able to measure this is the result of many, many years of work. This technique is called optical mapping, and it is almost 50 years old. In the 70s, the first measurements were done with voltage-sensitive fluorescent dyes, and people demonstrated that it is possible to measure action potential waves. And then this technique really had its heydays in the 90s, where people using a single camera were able to provide the first measurements of ventricular fibrillation and these wave phenomena in hearts, in isolated hearts. However, they performed these measurements with pharmacologically contraction-inhibited hearts. And in the past 10 years, I and other people have contributed or developed algorithms with which it is possible to track the heart and extract these signals from beating hearts. And so there are, I would say, 99% of all publications that were done, many thousands of publications were done with these pharmacologically contraction-inhibited hearts. And since 2016, 17, roughly, there are now more and more publications coming out where this compound called blebistatin is left out of the experiment, and we can now study for the first time mechanics and electrics at the same time. And also, what is kind of rare in our field is that people use multi-camera systems, such as our system, in order to reconstruct the 3D surface of the heart. So right now, as far as I know, this measurement that I showed to you is the only one of its kind. This is now a measurement of ventricular fibrillation. You can see that the heart is moving very slightly, almost not at all. And you can appreciate here also these chaotic wave patterns that we can observe now across the surface of the heart. They are spiraling, they're rotating, and that now begs the question, what is this? What are these dynamics? What is cardiac fibrillation? This is a very old question that has fascinated many people for many, many years. And in the 60s, a chemist, a physicist and chemist with who I also worked in Göttingen in Germany, came up with this hypothesis. Okay, I know that in the so-called Belousov-Zhabotinsky chemical reaction, I see these spiral waves, and these spiral waves must be related to cardiac fibrillation. Spiral waves are ubiquitous in nature. There are many, many different examples in either biology or in chemistry. This is here an example where I saw them here in the wild in California. And there are many, many other examples where you can observe spiral waves in nature. And these are all reaction-diffusion systems. Turns out they have all very similar equations, different compounds that become depleted or regenerated, and that creates these patterns. So how can these rotating patterns be created, or how do they emerge in the heart in particular? There were very early experiments, roughly 100 years ago, where somebody cut out a piece of a heart and stimulated in such a way that they were able to see rotating waves circulating in this ring of cardiac muscle tissue. And that was for a very long time then the central hypothesis. There must be these circuits in the heart that sustain this persistent activation. But since Valentin Krinsky, we also believe that this happens here in this tissue. So if you activate cells in such a fashion here that there's an open end of the wave, that can emerge in the real heart. Or if you keep this part here temporarily refractory, activate this part, then this following happens. This wave is able to rotate around its open end. And this is a spiral wave and the central mechanism in the heart that drives heart rhythm disorders, tachyarrhythmias. This here is a cardiac cell culture where you can appreciate that this wave pattern really exists in 2D at least. And this is a recording made by Flavio Fenn and Ilya Usulak at Georgia Tech. They really see here a voltage and a calcium rotational wave that is stationary and then degenerates into more complex wave patterns over time. These wave patterns are surprisingly robust. They can be sustained in highly anisotropic and very complex geometries. You can see despite in the atria, there's varying fibers and complex geometry, but this rotating wave can live on such a substrate. Depending on the parameters and depending on your genetic makeup and so on and so forth, physiological conditions, these waves can have all sorts of different shapes, and that is illustrated here in this 3-variable model. Depending on these 2 parameters alone, you can create various different dynamical regimes of these waves. Then the mechanisms how these waves can break up is manifold. There are many different mechanisms, but one is, for instance, that these waves can interact with themselves. This leading edge here crashes into the tail of its own arm and then can break up this wave. And this is also shown here. There are these breathing instabilities that can cause then further down the road a cascade into chaos. This is another illustration. This wave propagates here through tissue that is damaged. The white tissue is not supporting this wave that well, and it accordingly can lead to heterogeneity and breakup. So heterogeneity in general can promote arrhythmias. And now in 3D, it becomes a little bit more complicated, but even in entirely homogeneous media without any heterogeneity in the substrate, you can have a route into chaos. You can see here a scroll wave, and the blue line is a filament of the scroll wave. That's the center around which this wave rotates. And due to certain instabilities, these filaments can interact with themselves and break up and then cause such a complex turbulent wave pattern, which is also the underlying mechanism for this wave pattern here on the right side. So now to the part where AI and data-driven modeling can help us to better understand or diagnose heart rhythm disorders. We have applied various generic deep learning techniques, such as UNet, for instance, to solve certain problems in our field. One application, for instance, is that people are interested in localizing these regions here around which these waves spiral or rotate, because you can imagine that if you would remove this point, then this center here would stop shooting out these waves. And accordingly, this part of the heart would go maybe eventually back into sinus rhythm. So getting rid of these points, of these phase singularities, is a way of defibrillating a heart. And therefore, automatically tracking these points is important. And it was already shown in the late 90s that you can automatically identify these points. You embed this temporal signal using a Hilbert transform. And then in these resulting phase maps, you can compute the gradient of the phase. And whenever you compute a circular integral over the gradient around such a phase singularity, you get a discontinuity. And these discontinuities can then serve as a detection mechanism to track automatically phase singularities through or across the tissue. However, in practice, it is not easy to do that. Even with high resolution data, due to noise and other artifacts, it is sometimes you end up with spurious phase singularities or measurements that are simply not robust. So you can see here in a computer simulation, this works pretty nicely. But if you imagine you work with catheter mapping and you have sparse measurements only here on these points or along these points, then the underlying measurement is far from the ground truth, and accordingly the trajectories of these phase singular points they can deviate quite significantly, here shown on the right side, from the ground truths. So we thought this is a perfect problem for a UNet or autoencoder or LSTM version of a UNet to process such data. We trained such a network here, an encoding decoding convolutional neural network, with a sequence or a video of such data. So this here on the left side are voltage sensitive measurements, and then on the right side we either target discrete points directly, or we compute phase maps, and in these phase maps then phase singular points. And we saw that this network can do this easily with only 5 snapshots, 5 measurements from the activity. Even sparse measurements can provide you a phase map. So it can successfully cross-predict these phase maps from electrical measurements, and they are not far away from the ground truth. And they can do that with less than 10% of the spatial information. And so that showed us immediately how powerful neural networks can be for certain applications in our field. And you can compare that previous success or measurement with maybe this example here. Once you've seen this image here on the right side, it will also be much easier for you to anticipate what this sparse measurement of that image might be. And then what the network here does is it has seen so many examples of these dynamics that it can easily recover the underlying dynamics from it. The overall prediction accuracy, of course, goes down. The sparser and the more noisy the data gets, but overall we would still be able to identify here these phase singular points and therefore these rotational patterns across the surface of tissue. We have then applied it to our optical mapping recordings. You can see a ventricular fibrillation in a pig heart, and we can train this network either on this data or on simulation data, and then instantly compute these phase singular points. If we were to compute these phase singular points from our recordings, we would need a very long, much longer recording, at least maybe 1 second, whereas over here, only a few milliseconds are sufficient to compute these points. Most interestingly to us was that we were able to train it on simulation data, as shown here on the left side, and then apply it to experimental data, as shown on the right side, even though this data here, or these dynamics that are shown here, are 3-dimensional dynamics. And they are, what we see here is the surface wave pattern of a 3-dimensional wave pattern that evolves within the heart muscle. Over here, in contrast, the 2D simulations that we performed are really only 2D. So they're completely different dynamics, kind of. And of course, these computer models are also not fitted to this real wave pattern here, but nevertheless, it's sufficient for the network to perform relatively well. So you can see here that if we train on simulation data and apply it to simulation data, we have a very high accuracy, and we can train on pig data and rabbit data, meaning that we used isolated hearts from either a pig or a rabbit, still get a very high accuracy. And if we only train on simulations and apply it to pig or rabbits, we still get a decent angular accuracy. The next problem that we then tried to address was the following. We can image currently with our fluorescent dyes the 3D wave pattern, or the projection of this wave pattern on the surface of the heart, but these dyes do not really penetrate. They do penetrate into the heart muscle, but the light coming out of the heart muscle cannot reach the surface because the heart is too thick. And therefore, we don't really know what's going on underneath the heart surface. And we do not have measurements of ventricular fibrillation in 3D. We have computer simulations, but not real data. And we tested now in these generic toy model scenarios how well a convolutional neural network would be able to extrapolate the 3-dimensional wave pattern from 2-dimensional surface measurements. So you can see on the left side a 3D scroll wave pattern that shoots out waves towards the surface, and on the right side you see whatever we can see on the surface. So we're kind of pretending this is our experimental setting. And then trained a neural network to predict from 2D surface measurements. So this here is the surface layer of this bike peeled off and fed into the neural network. And we used 5 time steps, so 5 of these snapshots. And the target then was the full 3D pattern. And this here is a reconstruction. So you can see that works pretty well. When we look at it from above, you see this, the superficial layers, they are all reconstructed pretty well. But then if we zoom in and look at it in more detail, we see that the reconstruction accuracy decreases with depth. So the surface layers here, they are sharp and crisp and also accurate, but the deeper you go, the more it fuzzes out and becomes less accurate. We use diffusion models in the same way, training them to predict either from only the surface, top surface, or from the bottom surface, or from both surfaces, the three-dimensional wave pattern, and saw that they perform similarly to a simple unit architecture. However, if you provide them with more data, they can perform much better than unit. So we did a mistake, kind of. We tried to reproduce the same results as that we obtained with the unit. And with unit, we saw that there's a saturation. If you provide unit more than 5 frames, let's say 10 frames, 20, 30, 50, the network doesn't become better at it. And we then also stopped with the diffusion model with 5 frames because we thought, well, it didn't get any better with the unit model. But here, Stenger et al. Were able to show that if you actually use more data and provide more data to the diffusion model, and with more data, I mean, these 5 samples that we extracted were 5 snapshots of one rotation of such a spiral wave. Of course, you need to take into account how fast do these waves propagate to the surface and so on and so forth. And here, if you have much, much more data that goes maybe beyond one period of the spiral wave, then you can actually decrease this error quite a bit. Nevertheless, you can see that the deeper you go, the greater the uncertainty and error becomes, and therefore this is maybe not the best approach to try to extrapolate these complicated chaotic dynamics from surface observations alone. And therefore, another approach is needed. And this is something that I would discuss now in the second part of the talk. Before I jump into that, I wanted to mention that Eindhoven here in 1903 came up with this invention, the electrocardiogram. And since then, the electrocardiogram has had tremendous success in cardiology. Everyone uses it. It's very cheap. It's very effective. And you can immediately, even at home yourself, can assess the state of your heart. You can say, okay, my heart is beating normally, or oh, something is wrong. I need to see a doctor. And it's very cheap. So therefore, I think people have never really dared to come up with alternatives to this. The problem with the electrocardiogram and all electrical measurements is what I just showed to you. You cannot access the entire heart, and you cannot really visualize what's going on beneath the surface of the heart. And this focus on the electrocardiogram has maybe kind of blended us and prevented us from looking at what's already there. The heart beats, the heart contracts, and it's visible. We can use an ultrasound machine, we can use MRI, and we see the contractions of the heart and we can analyze them. And so therefore we could now ask the question, can we actually calculate the electrophysiology or the action potential wave patterns that trigger the contractions of the heart from the motion of the heart? And this is here now illustrated in these 3 idealized ventricular, biventricular electromechanical simulations. You can see on the left side, this plane wave here induces a very distinct contractile pattern that is distinct from the contractile pattern that is induced by 2 rotating waves or by 5 or more rotating waves. So it should be possible to compute something like that. But we didn't have the measurement data that could actually give us this certainty or this experimental evidence that would support this hypothesis. And here in our setup, we have now such data that motivates this even further. You can see here this wave that propagates across the ventricles of a rabbit heart, and this wave induces a contraction that is highly correlated with the action potential wave. It's not that everything contracts at once. It is really that the contractile activity sets on and follows the action potential wave. And then the 2 questions that we had initially, and we still have, I'm trying to address this, is this sufficient information to compute electrical activation? If you look at this mesh here, is that enough? And is this mapping between electrics and mechanics unique? These three images here show you how closely correlated that actually is. The action potential wave here propagating from the left to the right induces contractile activity over there. And because it contracts over there, which is also shown here in the strain rate image, tissue is being pulled towards the wave, and that tissue dilates as a consequence. And this is a very characteristic phenomenon that we observe over and over now in our experiments. And this wavefront here between dilating strain and contractile strain is following the action potential wavefront. There's a small delay that is typical for cardiac tissue, and it propagates in the same direction. So there is really experimental evidence that justifies this approach. And there are more observations that we made in the past. So here you can see waves with this is a recording that we made with a phone, mechanical waves that propagate through the heart. This heart was treated with cardioplegic solution, and therefore the activity was pretty slowed down, but you can see these wave phenomena. This now is the paper that I mentioned in the beginning. I studied this mechanical deformation pattern in the ventricular walls in computer simulations, and then also made recordings measuring here voltage, calcium, and strain. And you see here a clockwise rotating pattern in all of these three quantities. So down here, I was able to compute phase singularities that indicate the rotational centers of these waves for voltage, calcium, and strain. And we published then two papers that one is a physics-based approach, one is a deep learning-based approach, in which we try to compute now electrical waves for mechanical deformation. And I will briefly talk about this here, mainly because I wanted to highlight how much better deep learning works actually. So the first approach is a data assimilation approach. We say system 1 is the system that we want to measure. System 2 is a virtual replication of that system. We introduce sensors in the system that we want to measure and controllers in the virtual replication of that system, and drive the system with the measurement data that we have available eventually from system 1. So we can see here two excitable media, excitable systems, system 1 exists system 2, and there's a coupling term here. And this coupling term is really only the difference between the states of these oscillators over here versus here. And because videos sometimes explain that much better than 1,000 words, you can see that here. This computer simulation is coupled to this cell culture measurement over here, and is constantly receiving input from these sensors over here by the controllers that are placed in this domain. And you see that this simulation co-evolves with the cell culture. And we adapted this principle and modified it a little bit. We know that based on our measurements, these electrical waves induce also mechanical waves. And therefore, it might be possible to do exactly this, slightly modified. Instead of having in your control term a comparison between the two electrical states, you can now compare either an electrical and a mechanical state, or the mechanical state of system 1 with the mechanical state of system 2, and modify the electrics in such a way that the mechanics of the second system match the mechanics of the first system. So that's shown here. We have sensors in our drive system, or the first system that we want to measure, which measure mechanics, strain, for instance, strain rate, and they modulate the controllers in the electrics of the second system. So here we have a spiral wave that induces mechanical contractions. Red is contraction, blue is dilation. We added some noise to it and then observed that and induced electrical excitation in these locations here wherever contraction occurred with a negative electromechanical delay. And you can see then that over time, this system becomes synchronized with the first system. And therefore, we have a reconstruction of the dynamics in the first system based on a mechanical measurement. And then we were relatively happy that this also works for very complex wave patterns. So you can see here a comparison of the reconstructed wave pattern from mechanics with the original wave pattern. The topology is very similar. However, there were quite significant differences still. So we were happy about an 80% reconstruction accuracy. But all of that was kind of killed with deep learning, because it worked right away much better using simply an autoencoder for this problem. So we trained an autoencoder and a UNet with pairs of mechanical deformation patterns with electrical excitation patterns that cause these deformation patterns. Here you see a spiral wave pattern that induces, together with muscle fiber anisotropy, this anisotropic spiral wave-like mechanical deformation pattern, a focal wave pattern, a more complex wave pattern, and we use 20,000 samples of such pairs to then estimate electrical waves from mechanics with unseen data. So the network architecture, again, was a simple encoder-decoder. We analyzed displacements, X and Y displacements in 2D, which could either be with respect to an undeformed mechanical state or any arbitrary mechanical state, or the previous mechanical state of the system. And saw that this works pretty well after a short period of training and after a few epochs. This is an example of 3D dynamics, very, very turbulent dynamics. These dynamics induce tiny mechanical deformations here, which were analyzed by the network, and this is the reconstruction. It's visually indistinguishable from the original dynamics, so the complexity of the wave pattern is really no limitation. And you can see here on the right side, those are vortex filaments that were computed either from the original wave pattern or from the reconstructed wave patterns in black and in gray, and they perfectly overlap. So now the question is, how is this applicable in patients? Can we use this to reconstruct electrical activity in a patient who is there in the clinic and doing such a procedure? Here you can see this heart is beating abnormally. This patient has ventricular tachycardia, and at the center is the tip of a catheter. So this person performs this procedure here of mapping the patient and spends many, many minutes, maybe a full hour, to obtain a 2D map of the electrical activity. But since we are measuring this at the same time, this ultrasound recording, we could instantly provide this activation map if we are able to compute it from the mechanics. So we perform computer simulations again, idealized bioventricular computer simulations, which retain muscle fiber architecture that is necessary in electromechanical simulations. This muscle fiber architecture determines how the heart contracts, but also how the waves propagate. Then one issue was that we needed roughly at least 10,000 or maybe ideally 50,000 training samples. How do you get that many training samples in computer simulations that are usually traditionally very expensive? So we used Sphinxes, which is a smooth particle hydrodynamics framework, because it provided us sufficient speeds, and we were able to compute one episode here in less than 10 minutes and could therefore, over the course of a few days, simulate enough training samples. And then this problem becomes the following here. We have a point cloud of three-dimensional motion vectors, which describe the motion and deformation of the ventricles. The target is a static voltage wave pattern. Again, we have many, many different examples of these pairs. And the unit that we then use is a sparse res unit. We use this Minkowski engine here to process unstructured grid data and convert it into a format that this unit understands. The input are 5 frames describing 5 states of the mechanical deformation state of the heart that could in principle be extracted from ultrasound recordings or MRI recordings, and the target is then the electrical morphology of the wave pattern. In the computer simulations, we varied 4 different electrical models, which all produce very distinct electrical wave patterns with 4 constitutive laws for the mechanics, and then had randomized initial conditions, model parameters, the geometries of the tissues, and either induced focal or reentrant waves. You can see here the different anatomical geometries that also have an influence on the fiber architecture, and created 1,000 different geometries plus fiber architectures, and then varied our initial conditions. So we, for instance, stimulated the heart at the bottom, at the front, or at the back in random positions, or induced these different spiraling wave patterns also in random positions. And you can see here how these different electrical models produce very different quantitatively different wave patterns. Again, these are 4 examples. This is maybe a more realistic simulation that would be more comparable to what's happening in a patient who has a VT episode, and this is a more idealized computer simulation. We also in space varied the model parameters, accounting for spatial heterogeneity, and overall varied randomized more than 15, I think, parameters, and also included scar tissue or heterogeneity. So here in red, these parts of the tissue did not contract or did not conduct. And then we were happy to see that this works. We can actually train such a network. This here is the ground truth electrical wave pattern that induced this mechanical deformation pattern that are shown on the left. This was the input to the network, and this is the prediction. It works for focal wave patterns. We can compute now these activation maps here from these a sequence of predictions of electrical wave patterns. This is an activation map that was directly computed from the sequence of electrical wave snapshots. And then from this mechanical deformation, we computed this sequence down here and this activation map, and it's very similar. And you can now compute from the mechanical deformation of the heart these electrical activation maps, which are important for cardiologists because sometimes they're interested in where do these ectopic beats, these additional undesired beats, come from. It works for reentrant wave activity also pretty well. And these are the different accuracies that we achieved. How much more time do I have? Do I need to run through the remaining slides, or?
How many more slides do you
I think
have?
it's 11 now, and I might run out of time.
Yeah, maybe you can wrap up.
Okay, I will try to wrap up.
Next 5 minutes. Yeah.
Mm-hmm.
Next 5 minutes. Yeah.
Okay, good. So this is here an example where the heart includes also scar tissue, and the prediction nevertheless works quite well. This is very important because most patients actually do have myocardial infarcts and accordingly heterogeneity and scars. And if this method wouldn't work in the presence of scars, then it would be useless probably. So now how do we translate that to the clinic? We showed in computer simulations that this works, but that doesn't mean that it needs to work with patients. We have tried to apply a network that was only trained on simulation data on such data here on the right side, and that obviously did not work. And our approach now is to generate training data in our experimental setup with isolated parts, and then see how well that could perform maybe on real patient data, or fine-tune a model subsequently further down the road with all of these different types of data. So this is the experimental data that we have. It is really high quality electromechanical data of hearts. We now stimulate the hearts in such a way that we have many, many different stimulation sites and the corresponding mechanical deformation pattern. This is here just the optical recording. The red vectors indicate the mechanical deformation, but we also have, of course, transmural mechanical data from the ultrasound. And this is shown here. This is raw ultrasound data, a segmentation that deforms based on the 3D motion tracking that we perform. This is a corresponding target then, on the surface at least, of the electrical activity. And we are currently in the process of fully automating these experiments in order to be able to generate 50,000 training samples, or many, many thousands of training samples. So that means that we need to be able to go into these recordings, pick a certain frame, and then extract the frames, reconstruct it in 3D, cross-register these two recordings, and extract here the corresponding data, and fuse the data together. We've seen in other examples that in terms of generalization, we can train the model in the simulation domain and leave out one of the electrical models, and it still works pretty well. We've applied it to significantly different geometries. It also works in those. We have borrowed data from collaborators who use finite element modeling instead of SPH modeling that we use to train the model, and it also works sufficiently well. And I think that if we improve our of a methodology, it should be possible to reconstruct overall excitation waves from mechanical deformation. Now, the big question is, how do we get that to work in our ex vivo imaging setup? And let me jump through the last slides here. We showed that, for instance, in these two-dimensional cell cultures, we can apply a model that was trained only on simulation data on these experimental measurements and get reconstructions that make us hopeful that this could actually work in practice. As an outlook, of course, we would like to use this data not only for predicting one thing from another, but also maybe to evolve and obtain a data-driven model of the heart based on this experimental data that we're able to generate now in our setup. So we've used diffusion modeling and saw that diffusion modeling can be used to evolve cardiac dynamics. We can generate shapes and waves that look like ventricles and reentrant circuits. And I'm sure that we will also be able to generate beating hearts, maybe even driven by the data that we have now in our experimental setup. So that adds now a third pillar to our set of tools that we have, the conventional computer simulations, and now maybe hopefully soon also data-driven modeling of cardiac electromechanics. So, and I hope I didn't exceed too much. Thank you very much for your attention.
Very nice, very nice. Thank you so much for the interesting talk, Jan. I enjoyed it a lot. Let's have a Q&A sessions. So, well, I first want to introduce you to Robert Blake at Lawrence Livermore. Rob, are you still there?
Yeah, I'm still here.
Oh, cool. So Rob has been, working on the computational model for cardiovascular system. We have some computational tool. Rob, do you want to introduce?
Yeah.
Hi,
Jan. I worked with Natalia for a long time and wrote all our songs.
Yeah. Right. Mm-hmm.
So we have Cardioid here that can do a whole bunch of cardiac simulation stuff you might be interested in. I also have Melody that lets you take anything off CellML and quickly generate code for it to run in Chase or whatever simulator you want.
Mm-hmm.
And I have a really cool data set that you'll want to get access to. We did a whole bunch of simulations of just ventriculars and then recorded pseudo ECGs. And then we're able to train a neural network to go from the ECG to transmural maps on the AHA 16.
Mm-hmm. Nice. Okay. Yeah, we should definitely talk.
Yeah.
Awesome. All right, let's have a Q&A sessions. If you have any questions, please don't hesitate. Just unmute yourself and ask questions. I can start. So, you said the measurement can be only done on the surface, right? In order to fill the gap, you relying on the computational model. But every patient has a different shape of the heart, probably. And how do you deal with that? I mean, it's building those computational model. One of them will take some time, and if you have a patient with a different shape of the heart, do you parameterize the heart computational model so that quickly change the shape of the heart and run the heart model?
Good question. I think I don't know. I would assume that from imaging data we would have the shape of the heart, and the question would also be how sensitive is the solution to the shape of the patient? Is it possible to use a generic shape that represents most patients or that adapts slightly to the imaging data? I'm not sure how the anatomy would influence potential solutions.
Those imaging data is going to give you the shape of the internal structure of the heart as well for each patient?
Yeah, sure. We would have MRI data or generally we have MRI data or ultrasound data. Even though ultrasound has a pretty low spatial resolution, it's possible to extract at least the global shape of the ventricles from it. With the atria, it's a bit more tricky, but That's definitely possible. You wouldn't be able to extract, you know, finer structures, especially on the endocard. And it's also sometimes not easy to delineate the organ boundaries, but overall, we can definitely see differences in anatomy across patients in ultrasound.
Got it. One of the techniques you showed was you know, only using the surface data, and well, I mean, first you use the computational 3D computational model, and then generate the data and train the unit, for example. And then once the unit is trained, you just, you know, get the measurement in sparse way. Or you had some particular shape of the measurement, the location in the space on the surface. One was the uniformness distributed, and the other one was some, how should I say, you know, the straight line to the center.
Yeah, I think you mean the catheter mapping data, right? That was a study. Can I go back?
Yeah, if you can go back.
Let's see.
To the slide. Basically, my question is how do you, right, like the circular lasso.
You mean the star shape at the bottom right,
Yeah,
right? The array one.
yeah, exactly. How do you choose that location? Is it somewhat flexible to choose those locations of the measurement?
This is completely determined by the manufacturer of that catheter. So the study here by Roney et al. She there, I think, either chose generic representations of which, like, these geometers are similar to these catheters, or they are directly somehow created from the catheter. I don't know. But there are either these pentarray catheters or these lasso catheters, and then also basket catheters. Also, just different types of catheters that are on the market. Some of them have up to 64, I think, electrodes, the basket catheters. Not all of them measure something at the same time, but they can sense which ones contribute to the measurement, and there are often many different electrodes that record at the same time. So that's where it comes from. And in our study, we just used something that looks like a star.
Got it. And I think in another point of your presentations, you talked about the training the unit, and then use the trained unit to you know, get the information about the inside, not just the surface. I mean, the measurement is done on the surface, but also the internal. Yeah.
Yeah, this year. Yeah, right. So this is, we pretend that this is a piece of the ventricular wall. It's of course heavily oversimplified, but we were simply interested is from a dynamic systems perspective, is it possible to reconstruct very chaotic wave patterns
In 3D.
In 3D from only surface measurements, which is a situation which we have in our lab. We can currently really only measure the outer surface, the epicard, but in principle, you can also measure from the inside, the endocard at the same time. So you have two cameras, for instance, facing each other, and in between is the heart wall. And in that scenario, you have two measurements like here on this slide. And then whatever happens within the heart wall, the question was, can we recover that from only these two surface measurements? And based on what we've learned in these two studies here, Stenger et al. and Lebert et al. From 2023, it is possible to some extent.
In this scenario, like once that network, the neural network is trained, after that is trained for the inference stage, do you assume that the whole surface image is available as an input, or only the, you know, the sparse data on the surface is available?
Here we assume that the entire surface is available because the scenario that we tried to recapitulate was optical mapping, so optical measurement. we added some noise to it and saw it also works with noise, no problem, but we didn't try out the sparse measurement. So that would be the next step to move to more realistic geometries and add sparsity.
Okay, great. I mean, we have some technology of enabling that actually. Once you train the like autoencoder or UNet, and then only we get the sparse data on the surface and then try to reconstruct the whole 3D output data. Maybe I can introduce to you.
Yeah, that would be great. Yeah.
After the talk. Yeah. Okay. Well, that is my question. I don't want to take whole time of the Q&A sessions. Is there any questions from audience?
I have a billion questions, but it'll go weeds.
Only one or two allowed.
How much luck have you had with phase singularities? Every time I've tried to track phase singularities in cardiac tissue, like it's super messy to do.
Yeah, absolutely.
We eventually, we just gave up and started like just tracking wavefronts and measuring them as like a 4D surface instead.
Yeah.
But yeah, like wander so much, and depending on like little tiny things, they're very sensitive to error, and they kind of go woo 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 wobbly all over the place, and then disappear and reappear. Like continuity of phase singularities has always been really difficult for us.
Yeah, absolutely. I agree. So there are multiple factors that influence that. So the noise and notional artifacts and optical recordings, at least, they can cause, phase singularities to jump or disappear or something. You have spurious phase singularities that are not real, that are just a result of the filtering. So we have a protocol, I can send it to you if you want, that works relatively well when the signal quality is good enough. So you can process video data in a way that you focus on the wavefront rather than the wave itself. Both way works, and you have to slightly smooth the data, and then you remove outliers from the normalized data. So what you want to do is you want to normalize each time series to amplify the waves, and then use the Hilbert transform or whatever you need in order to embed this signal and compute the phase map from it. And then what is very important is to smooth the phase maps using a complex filter. So you convert the phase maps into a 2D vector field with a normalized amplitude, like a complex number, and then filter that. And with that technique, we're quite like it's pretty robust. Of course, it doesn't prevent you from detecting phase singularities that are further down, further upstream in the processing pipeline created there, or just simply the fact that you're looking at a projection of a 3D wave pattern, and some of these spiral waves, they meander really strongly. They have a linear core rather than a point around which they rotate, and that can also cause issues. And there's some work by a group in Belgium that came out recently where they look at that in more detail, but it can get very messy, yes.
Have you ever seen an arrhythmia without a phase singularity, either linear core or point core?
I know data from Flavio Fenton where he has shown that, I think. Have I seen that in my experiments? I don't think so.
Okay, because we saw it all the time in simulation. It would happen, especially around scar structures, and like the whole phase singularity stuff just really kind of broke down because we're like, we can't find, we have to do integrals over like half the heart. We can't find the phase singularities.
Yeah. The linear core is really an issue, and that has also raised concerns whether this concept of a phase singularity even makes sense. So the people in Belgium, Hans Dirks and his people, they published something last year, I think, where they expand this concept of a phase singularity into different lines, and it gets also really complicated, highlighting that it is a complicated topic, more complicated than at least just points, looking at points. I think it's a nice concept to look at, but I would not overemphasize it to describe fibrillation. It's in the end, it was supposed to reduce complexity. If you look at a three-dimensional wave pattern that changes quickly in time, so a 4D wave pattern, it can become very, you lose track of it. It's just complicated and difficult to understand. And therefore I thought back then in the 2000s, it was thought to help people to understand what's going on. And in a way it helps because you can count the number of waves, you can determine the density of these waves, you can see how waves interact with each other, these filaments when they touch each other and break up. That's a nice concept. But other than that, I think we also need other tools to look at cardiac fibrillation.
Great. I mean, I see that you're using UNet or autoencoder type of approach. That is nice because it gives you some reduced latent space dimensions and bring the data size effectively reduced to the smaller dimensions. But have you tried to actually interpret that any physical meaning on that latent space, the reduced dimension, like in the middle of the UNet or the middle space in the autoencoder?
Do you mean if you've ever used visualization tools of what's going on in the latent space, or do you
Well, the
just
physical,
principle?
yeah, physical principle, like your underlying physics is electromechanical systems. There's I saw you presented some partial differential equations associated with it, and of course the data is, if you use the simulation, the data will be really high dimension also after the autoencoder it is reduced, and it's quite unknown what physical meaning those latent space has. Maybe you can put some meaning to it, physical meaning to it, associated with those electric mechanical system you have. Have you tried that?
No, we haven't
If
tried
not,
that. We try to kind of verbally, talk it out what happens in the latent space. Maybe here in particular with these phase singularities, which is I think a much simpler problem where you only have a scalar valued field that you transform into a complex field. You have these two numbers that define the phase angle. There, I would say, the latent space, what is it? It's that conversion, that you do with the Hilbert transform somehow locally. It's much more local, I think. And in case of the electromechanics, you have long-reaching elastic effects. So global motion, I don't know how it translates. Maybe different modes of the contraction or of the deformation of the heart that are in the latent space somewhere. I mean, it's pretty amazing when you look at these simulations here, that you can convert something like this vector field on the left into a scalar varied field with such a complicated geometry, relatively complicated geometry.
Yeah.
It's amazing and
Very interesting. Very interesting.
Yeah, I don't know what happens, to be honest.
Right. Well, in our group, we try to, there's a framework we developed called the LASTI, latent space dynamics identification, meaning that, so once you project those 3D motions, vector field into the latent space through the autoencoder, and you get some dynamics in the middle latent space, and we don't know what's the governing equations for that trajectory data. So we try to associate that with some sort of differential equations, and then try to understand what that differential equations, what kind of meaning it has. So yeah, maybe I can introduce that to you as well. Okay, so I have a to-do list.
Sounds great. Yeah, I would love to talk about that. Yeah.
Awesome. All right. Any other question from audience? Well, if not, let's thank our speaker Jan for the wonderful talk and the interesting talk. I learned a lot, and hopefully we can further stimulate the collaboration between Lawrence Livermore and UCSF.
Yeah, would be great.
Yeah.
I was very happy when I found out about this seminar. So it was recommended to me by Michael Gravy.
Awesome. Awesome.
Yeah.
Thank you. Thank you, Jan.
Thanks. Bye.
Bye.